Greetings. We are going to cover now blood vessels and the circulation. This is in chapter 19 of your text. Now, we've already discussed about the heart. The heart pumps the blood, and the blood is sort of the fluid that goes around. It's considered, in some cases, in the old terminology, it was a liquid tissue. But in sequence, heart is pumping the blood through the arteries, capillaries, and veins of the pulmonary and systemic circuits. The pulmonary circuit, you have the pulmonary arteries, lung capillaries, pulmonary veins. This will be sent from the right atrium and the right ventricle. Now in the systemic circuit, you have the systemic arteries, tissue arteries, systemic veins. This is sent from the left atrium, then through the left ventricle to the aorta. Now, when we talk about these circuits, I want to bring up one other point that's extremely important. It is the amount of pressure that is in each of the circuits. For the pulmonary circuit, because it is very close to the heart, and you have a lot of capillaries very shortly away from the heart, you have a lower pressure that is required. For the systemic circuit, you have a much greater amount of pressure that has to exist to propel the blood throughout the entire body, including the capillary networks that exist all over in the limbs, brain, abdomen, the basically abdominal pelvic area, etc. Now, efferent vessels, these are vessels that carry blood away from the heart, so they will be referred to as arteries. Afferent vessels are vessels that carry blood towards the heart. They are veins. When we talk about exchange vessels, this is capillaries. They're thin walls. They permit exchange of nutrients, dissolved gases, and waste between the blood and the surrounding interstitial fluid. Here you see the right atrium, right ventricle sending blood out to be oxygenated in the pulmonary circuit. We start off with pulmonary arteries, come to the capillaries in the veins, capillaries in the lungs, and then the pulmonary veins, which return now the oxygenated blood back into the left atrium, then the left ventricle, and then throughout the entire systemic circuit. Arteries and veins differ in the structure and thickness of their walls. Now, in both of them, they have three distinct layers. They're called tunicas. We have the tunica intima, the tunica media, and the tunica externa. The tunica intima is the innermost layer. It's mostly endothelium, underlying a connective tissue layer. In arteries, the outer margin contains a thick layer of elastic fibers called the internal elastic membrane. Now, the tunica media, if you kind of figured that out, it's the middle layer, but it consists of concentric smooth muscle tissue with some loose connective tissue. In arteries, the tunica media is separated from the surrounding tunica externa by a thin band of elastic fibers called the external elastic membrane. The tunica externa is the outermost layer. It is mostly a connective layer sheath. Arteries have some smooth muscles and elastic fibers even there. Veins contain some smooth muscle and more elastic fibers. That's the major difference. When you look at arteries, arteries are going to have a much greater amount of smooth muscle in the media, etc. And that is so that they can adjust the flow of blood. The veins are going to have much more of elastic fibers because of their need to basically be the blood reservoir, which we'll get into later. Now, one of the other things you're going to find is the vasa vasorum. This is the vessel of vessels. These are blood vessels, both arteries and veins. They are in the walls of the large arteries that supply nutrients to the smooth muscles and the fibroblasts of the tunica media. The term vasoconstriction. This is where the tunica media smooth muscles contract. The lumen, where basically the blood is passing through, its diameter decreases. In vasodilation, the tunica media smooth muscles will relax. The lumen, its diameter increases. So basically we have a blood flow that goes from elastic artery to muscular artery to arteriole to capillary, in essence the capillary bed, then to venule, to medium-sized veins, and then to the large vein. If you notice from these images here, you can see a comparison of both the tunica intima, the tunica media, and the tunica externa. Here, if you look at this, the lumen of the artery is a little bit smaller. The lumen of the vein is much, much greater. But you'll notice that the smooth muscle in vein, its thickness is a lot less. Now you'll also notice some of these wiggle lines here. These are the elastic fibers in the artery. Also, you'll notice the tunica externa modestly present. There actually are a few smooth muscles here. In the tunica externa of the artery, you see some more elastic fibers here. The basic point that I want you to walk away with is that in an artery, it has a lot more flexibility. It has an internal elastic membrane and external elastic membrane. It has a thick layer of this tunica media, which is composed of smooth muscle. You will also notice the presence of the endothelium here and the endothelium here. The veins are capturing or bringing back blood that is much lower pressure, than the arteries are handling. So the arteries have to have this capacity to alter blood flow, whereas the veins are more or less just being able to having the capacity to bring the blood back to the heart. Sounds like a simplification, but actually it has some application here. Here are the features of a typical medium-sized artery and vein. And you can see this comparison here. The tunica intima, there's an interelastic membrane that's present in the typical artery but absent in the typical vein. The external elastic membrane in the tunica media, it is present in the artery but absent in the vein. In the tunica externa, you have an abundance of collagen and elastic fibers. In the typical vein, though, you have collagen, elastic fibers, and some smooth muscle cells. Keep in mind the general appearance. A typical artery is going to be usually round with a relatively thick wall. It's going to have a smaller lumen because really that wall thickness is taking up some of the space. Typical vein looks somewhat flattened or collapsed, in part because it really can't hold up its a round shape. It on its own is a relatively thin wall and also a large lumen. Now, there are 5 general classes of blood vessels: elastic artery, muscular artery, arteriole, capillaries, venule. The elastic artery includes the pulmonary trunk and the aorta. They're capable of stretching and recoiling as the heart beats, and you have arterial pressure fluxes. So it would bulge out a little bit as the pressure is higher, and then as it begins to subside, it will basically recoil. The muscular arteries, these are have a thicker tunica media, more smooth muscle than elastic fibers. They control the blood flow to organs and skeletal muscles. The arteriole is a poorly defined tunica externa, and the tunica media consists of scattered smooth muscle fibers. It's basically sort of the small range delivery to the capillary bed. And in the capillary bed, you have an exchange between blood and the surrounding interstitial fluid. So you're going to find we're going to have three different types of capillaries. They all have the capability to transfer material back and forth, particularly fluids and small molecules. But as you will see in a little bit, we're going to talk about being able to handle the exchange or leakage out from the blood of larger molecules. In the venule, that's basically a collection site to collect blood from the capillary beds. And then, of course, we've got the medium-sized vein and the large vein. The medium-sized vein, the tunica media is thin. In the large vein, you have a thin tunica media again, but a very large lumen. And this will include the superior and the inferior vena cava. Now, as you can see in this comparison chart here, large vein, medium-sized vein, venule, okay? You'll notice the differences in tunica externa, media, and tunica intima. You will see on this side here, elastic artery, because it has to take the pulsations of pressure as the blood is sent out from the heart, okay? And then as this kind of damps down in its pulsations, now what you're going to do is have the next size, which is the muscular artery, that is going to alter its lumen size by that thick tunica media and be able to divert blood to where it's needed most and where it's needed less, okay? We also have a smooth muscular cells, a little bit of them, as well as an endothelium for the arteriole. You can see the several different types of capillaries. We'll go over those more in a few minutes. They all contain endothelial cells, and they do have a basement membrane. There are just variations in that structure that allow for them to deliver more materials. So let's talk about capillary structure and capillary blood flow first. These affect the rates of exchange between blood and interstitial fluid. The capillary is the smallest blood vessel, okay? It's the site of blood tissue exchanges of nutrient and waste. Don't forget gases also, okay? The types are depending, they have depending differences in permeability. First off, we're going to talk about the continuous capillary type. That is the least permeable. It has an endothelial lining that is uninterrupted, okay? So basically endothelial cell to cell to cell to cell all the way around. Some cells are held together by tight junctions. You see this particularly so when you get to the blood-brain barrier, okay? But they permit the diffusion of water, small solutes, and lipid-soluble molecules into the interstitial fluid. The big issue is that because of this continuous barrier, you will prevent the loss of blood cells, which is important, as well as plasma proteins. In other words, think about it this way. Very charged molecules or ions are going to have a hard time getting through. They may get through with some assistance by facilitated channels, etc. But if you're expecting large molecules, plasma proteins, blood cells, nope, not going to happen. Not going to happen. Now, in fenestrated, they're more permeable than the continuous capillary. Their endothelial cells contain pores. This is called fenestrae. These windows that are covered by a thin basal lamina, that's it. They permit the rapid exchange of water and basically peptides between the blood and the interstitial fluids. You will see this in several examples. The choroid plexus of the brain, the capillaries of the hypothalamus, the capillaries of the hypothalamus, pituitary gland, pineal gland, and thyroid gland. Now, it may have sounded repetitious there, bear with me. There is something to think about here. You see, if you notice, I just mentioned 4 different endocrine organs. So this is what you're going to see primarily around many of the endocrine organs because you're going to have the release of hormones. Okay, they may be releasing hormones, stimulating hormones, or in a couple of cases, they're just basically the straightforward hormones like the pineal gland and the thyroid gland. But with the fenestrations, those molecules can pass through the membrane and get transported throughout the circulatory system. You will also see this in absorptive areas of the intestinal tract and filtration sites of the kidney. In sinusoids, they are much more permeable. Why? The endothelial lining is highly irregular and loosely arranged. Many pores and spaces between cells exist. This is a discontinuous endothelium. In other words, what I'm saying to you is, yes, folks, there are actually larger gaps between the blood and the tissue. This is going to allow for a free exchange of water and solutes and large molecules, some as large as plasma proteins. Where do you see sinusoids? The liver, the bone marrow, the spleen, many endocrine glands, pituitary, adrenal. Just one side note here, the spleen. As we've mentioned before, the spleen is involved in the processing of old red blood cells. Well, how are you going to get the old red blood cells out of the circulation system unless you have actually a gap in the capillaries to allow them to pass through? Okay, that's just to help you to make some connections here. When we talk about the term microcirculation, this is the circulation of the smallest of the blood vessels. When I talk about a capillary bed, that is an interconnected network between arterioles and venules. When I talk about the term thoroughfare channel, that's a flow that's relatively constant. The capillary flow may vary depending on metabolic demands of the tissues. To help you understand this, you're going to have some of the precapillary sphincters, which will pop open and close. And they do this with a highly sophisticated regularity. What do I mean by that? What triggers them to open up is the buildup of carbon dioxide, the buildup of waste. When they pop open, you have the levels reduce in the tissue, and then they can slowly start to shut down again. They are controlling, in essence, blood flow through these tissues that may or may not be metabolically active. Okay? So precapillary sphincters are bands of smooth muscles at the entrance of the capillaries. When I talk about vasomotion, this is somewhat what I was talking about earlier, this rhythmic change in the vessel diameter. Now, collaterals are multiple arteries supplying the capillary bed. When I talk about an arterial anastomosis, this is the fusion of two collateral arteries that supply a capillary bed. Okay? And when we have an arteriovenous anastomosis, that's a direct connection between the arteriole and the venule. To give you a concept that'll help you, when we talked about the heart in a previous lecture, we had an arterial anastomosis in some cases at somewhat the bottom part near the apex of the heart, where one of the major coronary arteries went down into a smaller one, and it directly went into and connected up with other arteries. Collateral circulation is the alternative circulation around a blocked artery or vein via another path. And this can be by nearby minor vessels. You will see it also in the circle of Willis in the brain, because there are other ways depending on where the blockage is. The blockage, don't be mistaken, can also affect behavior and affect brain function. But in some cases, the blockage might be there with bypasses that keep the brain well functioning and oxygenated. Here's an example of a continuous capillary. If you see here, here's the basement membrane totally covering it. Here are the endothelial cells, and they are going from edge to edge, cell to cell. Now, there's a boundary between the endothelial cells. In some cases, this is not going to be an easy boundary. It could be with a tight junction here holding it together. So how do we get substances from lumen to the outside tissue? That's where you're going to have vesicles containing materials transported across the endothelial cells. So you have first endocytosis and then exocytosis. Now, take a look here with the fenestrated capillary. You'll notice where are these thinning areas here. These are the fenestrations. All you really have here in these little spaces is the basement membrane. They're almost like little windows that allow for passage of much larger molecules. This is the boundary of the endothelial cells, so you would not see tight junctions or a real tight association of a boundary here. Therefore, you would have larger transport of larger molecules. Okay? Now, we go from there, we go to the sinusoids. And the sinusoids look like, what the heck is this? Notice that there is a gap between adjacent cells. There's a gap over here. A lot of times you'll also have some of the basement membrane there, but other times not. It's almost as if this was put together almost like a Lego set or something, but they've got big gaps in there, and that's because molecules can pass through. This is an example of an image of a capillary bed. Notice that this is a light microscopy image set at magnification 125. Here you have the small artery, and there you have the branching off into, from the arteriole into smaller capillary beds here, as well as capillary beds throughout here. Okay? Now, going over a few other things. Here are the arterial collaterals. They come together. Okay? And you have an arterial anastomosis. So basically, you've got several arteries supplying this one area for blood. Okay? And here's the tissue, obviously. Here's a thoroughfare channel that is really just a bypass. If none of this was metabolically active, all of these precapillary sphincters would close. And so blood would bypass through this from artery side to venule side. But you'll notice the differences in the key. A straight line is a consistent flow. So we have a consistent flow here. When we have sort of the staggered or the chopped up line there, what we have are really a situation where you can have a variable flow from one side to the other, in part because if there is serious metabolic activity here, you're going to have a need for glucose, you're going to have a buildup of carbon dioxide, and metabolic waste like urea, ammonia. You will also have greater need for amino acids, and you will, of course, have to need oxygen. And so this will all pass out of the capillaries. This will be triggered by opening of the precapillary sphincters right through here, okay? Here is also an arteriovenous anastomosis, as you can see here, okay? Now, let's talk about the venous system. Now, a lot of the force or pressure of the blood has been relatively high as we've gone through these elastic arteries and muscular arteries, but a lot of the pressure drops as you have all of this blood spread out through all the many capillary beds. Therefore, a lot of, if I can say it, the pressure energy has been lost. The arterial system has high blood pressure, but as you have the pressure changes that occur as you push the blood through the miles of capillaries in all your tissues, you end up losing a lot of the pressure difference. So the venous system is always going to be low pressure. As a result, also, it will contain almost 2/3 of the body's blood volume. Now, peripheral venules, these are, you'll see the blood pressure about 10% of that of ascending aorta. The pressure continues to fall along the venous system. Now, blood pressure in venules and medium-sized veins is so low that it can't overcome gravity. Wait a minute, doctor, what are you saying here? If you had basically none of the valves that are folds in the tunica intima in the veins, particularly in the lower limbs, you would not be able to get all the blood back into the inferior vena cava. Okay? You have to have valves that are going to prevent the backflow of that blood. And as a result, the veins are going to bulge a little bit. Now, if one is always in certain situations where they don't have much leg movement, but they're always standing, they succumb to a higher risk of varicose veins. This is due to the blood pooling in the veins, and the vein walls will become distorted. They're stretched out too much, and therefore the valves are not going to effectively close. Well, how do we have the valves do what they do anyways? This is where you get into the leg muscles. And as the leg muscles contract, they will squeeze on some of the veins, pushing blood up through one of the valves. But as the valves close, they prevent a backflow. Right? Now, if you stand all the time like a cashier, you're going to find that eventually one starts developing these varicose veins. Also, as a sidebar, hemorrhoids. These are varicose veins in the venous network of the anal canal. This can happen with various individuals. You may have seen situations where everyone is standing at attention. Maybe it's because they're in a military, a military setup, military parade, whatever, military display, and eventually somebody passes out. They fall face forward. Now, a lot of times what that happens is that the blood pressure has dropped. They're not periodically learning the trick. And the trick is to, even though you're not moving, you periodically will contract your legs, the muscles in your legs, in your thighs and in your calves. And as a result, you're pushing blood back up into the abdominal cavity, breathing a little bit, taking in deeper breaths, etc., because there is also in the thoracic area the capacity, because of the pressure changes in the thorax, to push the blood up and get it up to the heart. The individuals that don't know that trick, they find that if they've been standing for hours on end, the blood eventually is insufficient in its return. It's still there, they're not bleeding or anything, but they don't get sufficient blood delivery to the brain and they pass out. They hit the floor. Here's an example of what I mean. Here we have a muscular pump. This is the valve close. This is the valve close. Notice that when the individual contracts, they put pressure on the vein walls. This squeezes and pushes the blood up to the next area. And the valves superior are going to basically pop open. The valves that are inferior in the lower part here are going to be shut closed by the pressure here. Okay? Now, they're thinner walls, so they are going to be under more influence as opposed to, let's say, arteries in the leg. So if you basically just keep contracting the muscles, you're going to push the blood up, higher up into, eventually getting up into the abdominal cavity, etc. Now, this is where we get into what we call the muscular pumps. Okay, so basically the venous system is assisted in venous blood transport by valves and skeletal muscle contractions. Blood reservoirs. This is due to the veins being distendable, in other words, expandable than the arteries and veins, and they tend to hold more blood. Now, there's another interesting point here. In situations of hemorrhaging, such as profuse blood loss, the venous volume is reduced via the sympathetic nervous system action on the medium veins by venoconstriction. This reduced lumen diameter reduces the blood volume shortage. In other words, if I lost a liter of blood, that's a crisis. If I go to 2, it's even more of a crisis. But to compensate, what the body will do is it will start to push for venoconstriction so that more of the blood gets delivered back to the heart, and thereby in the circulation, And that's where you have individuals who've lost some blood but have not passed out, can still function for a bit. All right. Here's a pint of blood. Now think about this. When we talk about the different circuits, systemic, pulmonary, what blood is in the heart, systemic arterial, systemic capillaries, look at the difference here. This is like almost 2/3 of the blood volume occupying in the systemic venous system. That's going on all the time. Here's the situation when I was talking about venoconstriction. You have smooth muscle here. You have sympathetic nerves, and when they get activated, they are going to cause the smooth muscles to contract. Notice that the lumen goes from a certain diameter and gets reduced. As it gets reduced, the blood gets pushed farther faster because it's not going to have any time to hang out in the venous reservoir. Now we have to get used to a couple of concepts here. Pressure, resistance, and venous return will affect cardiac output. For those of you that forgot what cardiac output is, that's heart rate times stroke volume. And basically, in 1 minute, the number of times your heart beats times each stroke when you have the contraction ventricularly, how much blood is being pushed out. That will tell you the amount in 1 minute of blood that is being pushed out for circulation, cardiac output. Now, you want to keep in mind that both cardiac output and the distribution of blood with the pulmonary and systemic circuits must be constant. And it has to be constantly adjusted to meet the demands of active tissues. I broke that into almost like two thoughts there. One, yes, you've got to have this constantly occurring. Two, it has to be able to handle the dynamic changes that occur internally and externally. Gee, that sounds like homeostasis. Well, that's exactly what it is. Because think about this. There's times you're sitting right there listening to my lecture. There's other times that you're out, you're playing, you're running, you're jumping, you're playing basketball, you're riding a bicycle, you're walking around with friends. And so you have greater demands metabolically on some of the skeletal muscles. You're having a meal. You have greater demands on the digestive system, greater demands of its metabolic activity. So blood has to be shunted to different areas. And that's why you have this flexibility due in part to the arteries and the veins and the capillaries. Okay, so let's look at the sites of adjustment. First, we talked about cardiac output. You can increase your heart rate, and thereby you will also increase the possibility of more cardiac output. Arterial pressure. These are the forces exerted by the blood against the systemic artery walls. Peripheral resistance. The resistance of arterial system. This is opposed movement. Now, how do I get resistance? Well, if you remember, we can talk about a variety of ways of resistance. One of them is just increase or decreasing the lumen where the blood is passing through the capillaries or passing through, the arteries. Okay? Now, capillary pressure. This is the force exerted by blood on the capillary walls. We have venous pressure. This is the force exerted by blood within the venous system. And then venous return. This is dependent on the venous pressure, but it's the amount of blood arriving at the right atrium each minute. When we talk about capillary exchange, it's the process of diffusion between blood and the surrounding interstitial tissue. So all of these are going to play a role in being able to, what we would call in real time, minute by minute changes to adjust the blood flow to the metabolic needs of the different areas of the body. Cardiac output would play a role in affecting arterial pressure. Pressure would be also influenced by peripheral resistance. If I vasodilate, my peripheral resistance drops. If I vasoconstrict, my peripheral resistance increases. But the pressure changes can also influence the capillary pressure and thereby the capillary exchange between blood and the interstitial fluid. Then we have also the return, which is the venous pressure, and the venous return, which plays a role in cardiac output. All of this is controlled either by autoregulation or central regulation. What I mean by autoregulation, this may be situations within the tissue, but it may feed back also to the brain. The brain may play a role both in the control of peripheral resistance as well as venous pressure, as well as heart rate. And also, we have several other factors such as the adrenals with norepinephrine and epinephrine. Remember that they play a role in vasodilation or more vasoconstriction with these hormones. Some of them will play a role in vasodilation in other situations, such as sympathetic neural stimulation for blood flow to the skeletal muscles. And then we have also the kidneys, which would have an influence on several factors. One, the thickness of the blood. Well, listen, if I filter out more water, I end up with a greater solute concentration of the blood. That includes both formed elements, ions, plasma proteins, etc. If I choose to filter a lot less of water, I will have an influence on the viscosity and making the blood more fluid, reducing its viscosity and thereby increasing its flow. I hope that wasn't too complicated, but we'll go through all of this. This will make sense in time. When I talk about vessel luminal diameter, that's one of the main sources of resistance with the cardiovascular system. We have TPR, total peripheral resistance. This is the resistance of the entire cardiovascular system, but this depends on three factors: vascular resistance, viscosity, and turbulence. I basically have hit just about all of them when I was talking about the last slide, but let me bring this into more detail here. Vascular resistance. This is the opposition to blood flow in vessels. This makes up the largest component of TPR. Primarily, this is due to the friction between blood and the vessel walls. The friction depends on two factors: vessel length and vessel diameter. The greater the diameter, the lesser the amount of vascular resistance. The longer the vessel length, the greater amount of vascular resistance you're going to have. All right? So, the friction can be influenced by these two factors. All right? Let's think about viscosity. This is the measure of the resistance to flow caused by interactions among molecules and suspended materials in a liquid. What do I mean? Take a look at water. There's a very low viscosity, 1.0. Take a look at molasses. If you've never seen molasses, it is a basically very hypertonic fluid. It is a fluid, but it's very rich in pigments and sugars and things like that, okay? And it moves real slow. Its viscosity is labeled at 300. Can I have that in blood? Sure. If you end up with a polycythemic situation where you have a higher amount of a hematocrit, that's going to increase the viscosity and thereby increase the vascular. It's going to play a role to a point on this resistance. It's going to play a role on cardiovascular stress. It's just going to be harder to move. Think about how easy it would be to move if blood was just water. But instead, it's got formed elements and it's got salts and proteins and all these other things in it, okay? Now, turbulence. This is another thing that happens. Turbulent flow is fluid motion characterized by chaotic changes in pressure and flow viscosity. Please pardon my dogs in the background. Now, what goes on there? What am I saying? Basically, it's this. as fluid is moving through the center of the lumen, it's not going to have any problem. It's going to be pretty much in what we call laminar flow, sort of like a linear flow. But the fluid that comes up against the walls, they're going to have some instability and could be causing turbulence. Now, if you recall, we talked about formation of a plaque, okay? Arteriosclerosis. That bump in the artery will cause the fluid that comes up against it to create a lot of turbulence, and that's going to contribute to vascular resistance. Now, just to reiterate, turbulent flow is fluid motion characterized by chaotic changes. Okay, fine. This can also be caused by high flow rates, irregular surfaces, and sudden changes in vessel diameter. Okay, so you're going to have turbulence occur if you have to go from a certain lumen diameter and then you cut it down by 1/2. What happens with all of this turbulence? You see it, eddies and swirls, which increase resistance and they slow the blood flow. Just to help you there, if you've ever driven over or looked over at a river and it's flowing, and in the center it's nice and smooth, but if you look toward the edges, you can see how as the water is going up against the shoreline, you get these eddies and swirls and things like that. That's because in the center of that river, there's nothing that the water is being interrupted in. It's keeping itself at a certain flow velocity, nothing that contribute to chaotic changes in the flow. Nice laminar flow, straight flow. Where you have a shoreline, you're going to have some eddies and some swirls as the water molecules and the water flow gets bumping up against the edges and creates these swirling effects. Let's go over this from a diagram which may help. Friction and vessel length. The longer the vessel, the greater the reduction in flow. If I have a resistance to flow at 1, my internal surface area is 1. Fine, I've got a flow of 1. But if I double the surface area, I have increased the resistance to flow to 2. By doubling this, there's an inverse relationship here. Increasing your resistance decreases the flow. Okay? Here's another way to look at it. Vessel length versus vessel luminal diameter. The lumen is the area where blood is flowing, right? Okay. If the luminal diameter gets decreased, I'm going to increase the amount of contact of the flowing liquid, in this case blood, with the walls. I'm going to have a greater resistance to flow. So what I'm basically saying to you is, if my luminal diameter is large, I have a much less resistance to flow, therefore I have a speedier delivery of blood through the vessels. As I get smaller and smaller and smaller, I have a greater resistance to flow, and therefore less blood is delivered through that vessel. Friction and vessel luminal diameter, as you can see here, the greatest resistance is near the surface, right over here, against the edges. The least resistance is at the center. Okay? That's part of the key also. Here's this concept about viscosity. Real simple. If you're not familiar with molasses, think about it as motor oil. Think about it as maple syrup. All of these are thickened fluids. It takes a greater amount of energy to push them through any tubes. Okay? Blood is not water. Blood is much thicker than water, even though there's that saying that blood is thicker than water if you're not familiar with it. But anyways, blood has, remember, not just water. It is the plasma proteins, it's the formed elements, it's the thrombocytes. It may be other materials present in it, and as a result, it tends to have a thicker or higher viscosity, and thereby you have a greater amount of pressure that's necessary. If I doubled the amount of red blood cells, I would have a much harder time pushing the blood through the vasculature that we call my blood vessels. Here's turbulence. Let's say this is a plaque deposit right here and here. As a result, remember that you have the slowest movement or the highest friction as you come up against the wall. Well, the wall now here is reduced in size, that's one. But also, as water or any other fluid would come up against these bumpy areas, they would start having turbulence. In other words, they would start, instead of moving straight, they would have to follow through along this, and this would create turbulence or eddies or interactions, and so you don't get as much flow. The flow is reduced as you have increased unevenness of the walls inside. Okay, so let me just go over this with a little bit of, yeah, it almost looks like math. Don't panic. Blood flow is determined by the interplay between arterial pressure and peripheral resistance. Blood flow is the volume of blood flowing per unit of time through a vessel or a group of vessels. Blood flow is directly proportional to arterial pressure. Okay? So blood flow equals whatever the arterial pressure is. Blood flow is inversely proportional to peripheral resistance. So flow, and this is what PR is inversely. So it would be the opposite. The greater the PR, the lesser the flow. The greater the flow, it must be a decrease in the PR. Okay? When we have a pressure gradient, that's a difference in pressure from one end of the vessel to the other. Now, cardiovascular pressure is measured in millimeters of mercury. That's the mmHg. You want to take time to review vessels. And when you do that, the review is based comparing the vessel luminal diameter and the total cross-sectional area, the average blood pressure, the velocity of flow. Now, if you get messed up with this, here's a couple of quick points to help you. Remember systolic pressure, SV. That's the pressure that is the highest amount, and it's usually when the heart has pushed this blood out. Systolic pressure is the lower end, when the ventricle is beginning to now relax and the pressure thrust has stopped. Pulse pressure is the systolic pressure minus the diastolic pressure, and they call that PP. The mean arterial pressure is MAP, and MAP is basically the diastolic pressure plus 1/3 of the pulse pressure. Now, one of the things, if I was in classroom, I would basically be walking through this and saying, okay, in the elastic arteries, you have so much vessel luminal diameter. This is, of course, your cross-sectional area. This is your pressure, the vessel luminal diameter here. This is the average blood pressure. This is the velocity of blood. So what you're going to see is this. As you get going from just a few elastic arteries to more muscular arteries to lots more arterioles, and then you hit capillaries, and then you see this total cross area, sectional area. That's if you took all of the lumen spaces and added them all up for all the capillaries, etc. And then, of course, it drops down for the venules and the veins, etc. All right? Look at it from the perspective of average blood pressure. The average blood pressure right at the point of the aorta, there you are at your peak, or your average, and then it starts dropping down as you go into more and more from elastic arteries to muscular arteries to arterioles. And then you notice this great pressure drop from one end of the capillaries to the other end of the capillaries. Then it kind of has a different slope in the venules, veins, and the vena cava. Okay? The better way to look at it, if you get confused, is the velocity of blood flow in centimeters per second. At the aorta, wow, it's really speeding along there. And as you go from elastic arteries to muscular arteries and then to arterioles, the speed drops. In the capillaries, the velocity of blood is pretty much a flat line. That's because you're going to get the roulette where you're going to have the red blood cells all line up nice and happy, moving through the capillaries like little soldiers, boom, 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 having fluid exchange, etc. And then you start having less of these vessels because now you're collecting all the blood into the venules, into the veins, and into the vena cava. Keep in mind also at the point of the aorta, which will stretch out and recoil back. That's because you're going to have the ventricular thrusting of blood out and then the relaxation, and then the ventricle contracting, relaxation, contracting, relaxation. So there's going to be this pulsatile manner, but what you notice is that all the pulsatile activity pretty much dies down by the time you get to the arterioles, but you do note a major pressure slope here, going from one end of the capillaries to the other. And then the slope is much more gentle here, as there's very little change from venules, veins, medium-sized, large veins in the vena cava. Okay? Now, the capillary exchange involves three processes: filtration, diffusion, osmosis. When we talk about capillary hydrostatic pressure, CHP, that's the blood pressure within the capillary beds that provides the driving force for filtration. In other words, material leaving the blood going into the interstitial fluid. Size selection controls solute transfer. Huh? What's that mean? Simple. The bigger the molecule, the less chance you're going to have a solute transfer, unless you get something like sinusoidal. Somewhat maybe if you have fenestrated, but definitely not your common, average, everyday capillary. The bigger molecules, like albumin, etc., are not, and supposedly not supposed to be showing up in your capillary tissue beds. They stay in the capillary. Red blood cells, white blood cells, etc., they stay there. Here, capillary hydrostatic pressure. Here is your capillary. You have the endothelial cells here. You have a little gap here. So what happens? Small solutes, okay? Water molecules, they can pass through, okay? So you're going to have ions pass through. You're going to have water molecules pass through. Major blood proteins will not. Some amino acids might, or some amino acids will be assisted across by this passage through the endothelial cells. All right? Now, we're going to go through all the pressure changes, etc. Capillary exchange is a dynamic process. I told you before, vessels, the precapillary sphincters open, close. Open, blood rushes through the capillary. Close. Open, close, depending on the metabolic demands of the tissue at the time. We'll see this in the diagrams in a second. Near the arteriole, filtration is predominating because you have the CHP being greater than the BCOP. What's BCOP? Blood colloid osmotic pressure. This is basically the plasma osmolarity. What's CHP again? I forgot. Capillary hydrostatic pressure. In other words, what is the hydrostatic pressure, the pressure of the blood in that capillary? When it's greater than the BCOP, molecules will move out from the capillary, but the molecules are going to be also filtered. Some can pass, others cannot. Water can, dissolved gases like carbon dioxide and oxygen can. A red blood cell cannot. A albumin molecule cannot. Okay. When you get to about the middle of the capillary, this is where the CHP is equal to the BCOP. So there's no net fluid movement. Now, to help you understand what the blood colloid osmotic pressure is, that's the capacity of the colloids present. In other words, all the different, the thrombocytes, the proteins, the movement of certain molecules, the lipoproteins, antibodies, and all other large molecules, they're going to have this capacity to try to pull water back into the capillary. Now, when CHP equals BCOP, no net fluid movement. As you get closer and closer to the venule, though, you're going to start having reabsorption. That's because the BCOP will be greater than the CHP. After you've discharged some of the water and some of the dissolved gases and some of the smaller molecules, you've altered the CHP. It's dropping. The BCOP is staying where it's at, and eventually it gets greater. And as it gets greater, fluid now moves back into the capillary. Okay? Now, there is a concept called NFP, net filtration pressure. That's the difference between CHP and the BCOP. If CHP is greater than BCOP, then NFP is positive. If it's the other way around, where BCOP is greater than CHP, then NFP is negative. Okay? Net filtration pressure. Okay? The routes of diffusion across capillary walls, keep in mind the following. Water, ions, small organic molecules such as glucose, amino acids, urea, can usually enter or leave the bloodstream by diffusion either between the adjacent endothelial cells or through pores of the fenestrated capillaries. No problem. What else can move? Ions, sodium, potassium, calcium, chloride. They can diffuse across the endothelial cells by passing through channels in the plasma membranes. Okay. Large water-soluble compounds are unable to enter or leave the bloodstream, except at fenestrated capillaries. Those you would see in the hypothalamus, the kidneys, many of the endocrine organs, and the intestinal tract. What about lipids? Lipids, such as fatty acids and steroids, and lipid-soluble materials, including soluble gases such as oxygen and carbon dioxide, can cross the capillary walls by diffusion, again through the endothelial plasma membranes. Plasma proteins are normally unable to cross the endothelial lining anywhere except in sinusoids, such as those that you would see in the liver, where, interestingly enough, plasma proteins enter the bloodstream. The liver makes some of these plasma proteins. It's absolutely essential. Now, remember, CHP minus BCOP gives you NFP. If CHP is greater than BCOP, you have a positive NFP, which means fluid leaves the capillary, goes into the tissues. If BCOP and NFP are equal, then there's no real net fluid movement. If BCOP and CHP, where BCOP is greater than CHP, then you're going to have an NFP that's negative, which means that fluid is going to start rushing back to this side of the capillary. Keep in mind one thing that's very, very important. This is the arterial side, so you would expect more fluid to leave. This is the venous side, so you're going to have a recovery of fluid, but not an equal amount. Notice 24 liters here, 20.4 liters here. So what have you got? Oh, gee, I got about 3.6 liters a day. They're still hanging out here, but they're picked up by the flow into the lymphatic vessels. Okay? Now, how are you going to get variations in capillary exchange? If hemorrhaging occurs, because both the blood volume and blood pressure will decline. This decrease in CHP lowers the NFP and increases the amount of capillary reabsorption. The result is a reduction in the volume of the interstitial fluid and an increase in the circulating plasma volume. This is known as recall of fluids. In essence, what I'm saying to you is this. You're in a car accident, you lose a pint or two, or maybe a little bit more of blood. What is going to happen is you're going to have a reduction in blood volume, reduction in blood pressure. And so what's going to happen is that area of the capillaries, and all the capillaries, is going to have a reduction in CHP. That means you're going to have a lowering amount of NFP. You're going to have more fluid, not leaving each of the capillaries, but more fluid coming back from the capillaries. And that will be a compensation mechanism to help in the midst of a severe hemorrhaging. Now, what about dehydration? Wow. Well, this can occur also. The plasma volume decreases because water loss. The concentration of plasma proteins increases. That's going to push up the BCOP. Okay? And so you're going to have a much greater amount of reabsorption. All right? Now, this is an attempt to blunt the dehydration situation, but you can only get just so much, and you've got to start doing fluid replacement externally. If the CHP rises or the BCOP decreases, the fluid moves out of the blood in the capillaries and builds up in the peripheral tissues. And this is what we call edema. Cardiovascular regulatory mechanisms respond to changes in blood pressure or blood chemistry. This is very important. The homeostatic mechanisms regulate activity to ensure tissue perfusion meets the tissue demands for oxygen and nutrients. When I talk about tissue perfusion, I'm talking about blood flow through the tissues. Now, vasodilators, these are factors that promote dilation of blood vessels. Think about it. If I dilate the blood vessels, hey, I should be having more blood delivery to the tissues. There's another concept which we've kind of touched upon earlier. That's autoregulation. This is local changes in the pattern of blood flow within capillary beds. And these changes are due to the opening and closing of precapillary sphincters to control blood flow into the capillary beds in response to chemical changes in the interstitial fluid. Keep in mind, those chemical changes are key. And these are signals that's saying, is the tissue going through some metabolic activity? Think about it. If I take my muscles and I'm stretching and I'm doing whatever it is, playing basketball, lifting a shovel or something, I'm having chemical changes in that interstitial fluid in the skeletal muscles. Therefore, autoregulation is going to kick in, and it's going to demand that the precapillary sphincters open up so that capillary beds are well perfused, and exchange of nutrients and wastes is occurring. Here's where we bring up that magic word we've heard since the first chapter, homeostasis. First chapter of AP1, that is. Look at, when you have an adequate blood flow, local blood flow and blood pressure, the effectors, the free capillary sphincters, are going to respond. They're going to have local decrease in resistance and an increase in blood flow. Homeostasis is restored by increasing the blood flow. Okay. If, notice right up here, autoregulation does not restore homeostasis, then we start acting on these structures: kidneys, the endocrine response, the long-term increase in blood volume and blood pressure, the nervous system. We're going to have changes that are going to trigger systemic blood due to the changes in the blood pressure and the chemistry. We're going to act on the cardiovascular center. In the medulla oblongata, you're going to increase blood pressure by sympathetic stimulation of the heart and peripheral vasoconstriction. Now, there are baroreceptor reflexes. Baroreceptors, if you remember, those are receptors or sensory receptors that respond to changes in pressure. In this case, we're responding to changes in blood pressure. The arterial baroreceptors are located in the carotid sinuses and the aortic arch. The carotid sinuses, they're near the base of the carotid artery in the neck. And responses to increased baroreceptor stimulation, the cardiac centers are going to decrease cardiac output through parasympathetic stimulation and sympathetic inhibition. What I'm just saying is they're going to slow the heart down. Okay. You will have widespread peripheral vasodilation due to the inhibition of the vasomotor center of the medulla oblongata. What if I had a decreased baroreceptor stimulation? Okay, the cardiac centers are going to increase their cardiac output. And this is going to be via the stimulation of the sympathetic innervation to the heart. This results from the stimulation of the cardio-acceleratory center in the medulla oblongata and the accompanying inhibition of the cardio-inhibitory center. You will have also widespread peripheral vasoconstriction caused by the stimulation of the vasomotor center. If you were in a crisis, such as a major blood loss, the sympathetic activation occurs, both norepinephrine, epinephrine enter the bloodstream. The net effect is an immediate increase in cardiac output and generalized vascular constriction, which is a combination that rapidly elevates blood pressure. And here you see this all done up nicely in this diagram. I encourage you to review this chart. It is going to be very helpful, especially for my fine students that are in nursing and allied health, etc. Think about this. This is what you're doing now to get this down. Later on, you have the clinical situations that you learn from it. But the key point, if I lose blood, my blood pressure is dropping. How am I going to have my system compensate for it? At the same time, if I have an increasing amount of blood pressure, how is my system going to compensate to reduce the blood pressure? And this is all involving homeostasis. Now, what are the endocrine responses to low blood pressure and low blood volume? They're very different from those of high blood pressure and high blood volume. There are long-term and short-term endocrine responses to cardiovascular performance. Also, the responses involve kidneys and the heart in the endocrine responses. Let's start off. Drop in blood pressure and volume. Short-term response, epinephrine, norepinephrine. So we're going to end up with vasoconstriction, etc. Long-term, the response would be pump out more erythropoietin. That's going to lead to more blood cells made. Renin release leads to angiotensin II activation. You also have antidiuretic hormone release. Now keep in mind, antidiuretic hormone at a certain level, yeah, that's going to act on the kidneys. It's going to retain more water to increase the volume of blood. But if it really gets released more, it can also enhance a vasoconstriction. Okay? You can see how these are worked out. If I had a rise in blood pressure and volume, now in the heart, we would release natriuretic peptide, which inhibits ADH release, inhibits aldosterone release, inhibits the release of epinephrine and norepinephrine, leads the kidneys to increase sodium and water excretion in the urine, therefore your volume is going to decrease. It is also going to decrease the blood pressure. It will signal peripheral vasodilation, which further will decrease the pressure of the blood. Natriuretic peptide, as I mentioned to you in the endocrine section, is released by the stretch of the atrial or ventricular heart walls. The atrial is made in the atrium. There is the other peptide. They used to call it ventricular natriuretic peptide. They now call it more brain natriuretic peptide because it's made in the ventricles as well as in the brain. Just as you understand, they both act in the natriuretic effect, which is enhance water excretion via enhancing sodium excretion, as you can see right here. So increasing blood pressure and volume acts on the cardiac muscle cells. You end up with ANP and BNP, BNP being that type they call brain, but also it would act on the kidneys, the blood vessels, and you would see all of these eventual effects which lead eventually to decreased blood pressure and blood volume. Now, we also have chemoreceptors that monitor the chemical composition of blood and cerebral spinal fluid, CSF. Chemoreceptor reflex. This is a response that changes in carbon dioxide and oxygen and pH levels in the blood and the cerebral spinal fluid. The chemoreceptors are located in the carotid bodies, the aortic arch, and the medulla oblongata. I want you to note this. Remember that if you take carbon dioxide with water, you get the formation of carbonic acid. And carbonic acid is unstable, and therefore it will break down the bicarbonate ion and hydrogen ion. Now, this reaction is reversible, so you can take bicarbonate ion and hydrogen ion, bring them together, carbonic acid, that can break down to carbon dioxide and water. Remember that you also have an enzyme that will speed up the reaction called carbonic anhydrase. Okay? Now, here's a couple of pointers here. Let's say I've got increasing CO2 levels and either a decreasing pH, which means the blood's getting more acidic, and I'm also having decreased O2 levels. What happens? This is where the chemoreceptors come into play. You will see the carotid and the aortic bodies and the medulla oblongata all signaling. The effector will be the medulla oblongata. You will have the respiratory center stimulated to increase gas exchange. The cardiovascular centers, cardiac-acceleratory center is stimulated. The cardiac inhibitory center is inhibited. The vasomotor center is stimulated. What is this going to result in? With an increased respiratory rate, I'm going to increase the pH as well as the O2 saturation levels in the blood, as well as get rid of some of that carbon dioxide. When I work in the cardio-acceleratory and cardiac inhibitory center, I'm going to increase cardiac output and blood pressure. With the vasomotor center stimulated, I'm going to increase vasoconstriction, which will elevate the blood pressure. And again, all of these will lead to more blood delivery for gas exchange to get rid of carbon dioxide and to bring in more oxygen levels. And these factors will reverse the pH of the blood and move it back up to the normal range. The cardiovascular center makes extensive adjustments to cardiac output and blood distribution during exercise. We have the respiratory pump. Now remember you talked about the muscle pump. Let's talk about another way to get an enhancement in the venous delivery of blood, okay? The respiratory pump is a mechanism whereby inhalation creates a negative thoracic pressure that pulls blood into the vena cava from the branches. You want to keep in mind that the presence of the skeletal muscle pump in the limbs to help squeeze blood from the peripheral veins contributes also. You want to compare blood distribution between at rest, light exercise, and heavy exercise. As you can see here, when we have rest, you have a certain amount going to the brain. Notice that right across the entire chart here, you can't change blood delivery to the brain. That's a no-no. I know it sounds silly, but it's a key point that even in light, heavy exercise, etc., you always still have a certain amount of blood going to the brain. Now, notice, for example, skeletal muscle. When I go from at rest to light exercise, I have increased by almost 4 times the amount of blood per minute going to the skeletal muscles. And goodness gracious, by heavy exercise, I've gotten it up to about 10 times that, 12,500 milliliters per minute. Here's an interesting fact. Think about this. At rest, you've got 5,800 milliliters per minute of the cardiac output. Go to light exercise, it's 9,500. Go to heavy exercise, it's 17,500 milliliters per minute. So what's going on? What you have are a greater amount of cardiac output occurring, but now you're shunting blood to different areas. This brings us back to AP1 and sympathetic and parasympathetic systems. Remember, fight or flight? I don't need to digest my meal. I need to either fight or flee the threat. Okay? And by the way, those lower numbers here, this is your cardiac blood supply to the heart muscle itself. Notice you go from 250. As you start getting more active, you go to 350. And as you go to heavy exercise, you're now pushing 3 times the amount of blood through that heart muscle. Same thing, if I'm going sympathetic activation, I'm going to decrease the amount of blood going to the kidney. I go from 1100 to 900 to eventually 600. There's a decrease that's going on because, hey, I've got to have all the blood volume I need right now to fight the lions and tigers and bears, oh my. What you have to keep this in mind. Same thing with abdominal viscera. Under sympathetic stimulation, we're not going to care about digesting a meal. We go from 1400, then to 1100, then to 600. In other words, you got a progressive drop. Now, if it was parasympathetic activation, you wouldn't have, obviously, the exercise stuff. Yes, you would have more blood going to the abdominal viscera. Okay? Now, one interesting thing also to keep in mind. Skin, 500 at rest, 1500 at light exercise, 1900. Why? Thermoregulation. As you're increasing your exercise, you've got to get rid of excess heat. And the way to do it is to deliver the blood to the skin, which in part you're going to have some radiant cooling, etc. You're also going to be pushing out more sweat. Okay? Hope that helps. Now, let's talk about short-term and long-term mechanisms that compensate for a reduction of blood volume. This can get sensitive, so I want you to be understand that this is extremely important. I want you to review the short-term and long-term responses to blood loss. You need to be aware of shock, which is acute cardiovascular crisis marked by low blood pressure, hypotension, and inadequate peripheral blood flow. The shock develops when normal compensation mechanisms fail to maintain adequate cardiac output and peripheral blood flow. Circulatory shock involves a series of interlocking positive feedback loops that begin when the blood volume exceeds about 35% of the total blood volume. Progressive shock, it's an initial stage characterized by positive feedback loops that accelerate the tissue damage. Then we have also irreversible shock. This is fatal in the final stage of shock when the circulatory collapse occurs. Let's see what I mean here. Well, right off the bat, you've seen this before. Decreasing blood pressure, volume, this is caused by extensive bleeding. The responses by the nervous system, they're all going to be kicked off by the baroreceptors and chemoreceptors, and they're going to act on general sympathetic activation, stimulation of cardiovascular center, okay? And this is going to be the response by the cardiovascular system, peripheral vasoconstriction, mobilization of venous blood reservoirs, increased cardiac output. What about the kidney system? This is going to be coordinated by the endocrine system. Yes, you're going to see long-term hormonal responses, greater amounts of ADH, angiotensin II, aldosterone, and EPO released. This is going to elevate the blood volume. So, combined short-term, long-term effects, we have an increased blood pressure and increased blood volume. Okay, that sounds nice. These are the short-term responses. I expect you to know them. Straight out, I'm going to say it right there. No hint, hint. It's straightforward. Know them. Because of the fact that when you start increasing cardiac output, you start triggering peripheral vasoconstriction. You can see an increase in heart rate. You may not get as much of the cardiac output because you've lost some of that fluid. Long-term responses to blood loss, the decrease in capillary blood pressure triggers a recall of fluids from the interstitial spaces. Now you're going to have aldosterone and ADH promote fluid retention and reabsorption by the kidneys. Your thirst is going to increase and additional water is obtained by absorption across the digestive tract. You're going to pump out EPO so it can stimulate the maturation of red blood cells in the red bone marrow and resulting in increased blood volume and improved oxygen delivery to peripheral tissues. Okay, But this chart is the one that I want you to spend some time reviewing. Start off with 1. You have now severe reduction in blood volume. That means you're going to have reduced cardiac output. And reduced cardiac output is going to start having a decreased cardiac blood flow. And it's going to be a positive feedback that's going to lead to damage to the muscle. As you keep having this decreased blood flow, damage to the muscle, it's still trying to pump out blood. You're also going to notice that with the decreased cardiac output, you're having a decreased arterial pressure. Now, you're going to eventually have decreased peripheral blood flow, as well as you're going to maximize out, or max out, as you might say, vasoconstriction. Hang on, we've got more. Decreased peripheral blood flow, you're going to have an increased PCO2 and lactate and decreased pH. So you're leaving pH 7.35 to 7.45. You're going below that. Now that's going to increase capillary permeability. Now increased permeability means more blood volume is lost. The capillaries are going to have fluid leak out. Because what's also going to happen is with the increasing amounts of CO2 and lactate and the decreasing pH, you're going to start having clotting within the vessels. Clotting within the vessels is going to decrease peripheral blood flow, yes. It's also going to decrease the venous return, which means even more decreased cardiac output. So let's go down to the last area, which is the nightmare. You can max out on vasoconstriction only so long. Now you're having irreversible cardiac muscle damage. And remember, any cardiac muscle damage means that the efficiency of each stroke is reduced. And therefore, if you're having reduced in stroke volume, that means you're going to have a decreasing arterial pressure. And this is where we go into another loop. Decreased blood flow to the CNS, irreversible CNS damage. When you start having a decreased blood flow to the central nervous system, particularly the brain's going to be affected. As the brain is affected, you're going to have eventually a decreased sympathetic activity, which is now going to slip off, keeping all of the vasoconstriction working, keeping all of the arterial pressures up. It's going to start sliding down. As it slides down, you finally have circulatory collapse and death. Okay, moving on. New blood vessels form through vasculogenesis and angiogenesis. Well, what's the difference? Let's start with that. Vasculogenesis is the formation of new blood vessels by hematoangioblasts and precursor endothelial cells. The products are your dorsal aorta and cardinal veins. Now, angiogenesis is the growth of new vessels from pre-existing vessels. In essence, you've got to have vasculogenesis first, then angiogenesis. The dorsal aorta becomes the descending aorta. The aortic arches form the carotid arteries, the aortic arch, and part of the pulmonary arteries. The cardinal veins form the superior and inferior vena cava and the vessels returning to the heart. As you can see here, we move in from an embryo, and eventually you get to the point of a much more sophisticated embryo here. Here you start off in vasculogenesis. Some hemopoietic cells, remember these are going to make blood cells. These are hemangioblasts. Then you have a primitive capillary network. Then, after you've had the formation of the cardinal veins and you've had the aorta, you start having this buildup and more sophistication here. Mind you, also note this, and we'll get into this later on in the development chapter. Yolk sac. Everybody goes, well, wait a minute, I'm not a chicken. No, but the yolk sac in an evolutionary, a developmental evolutionary approach, the yolk sac exists still, but we're not birds. We don't hatch from eggs. So we don't have as much demand on it, but it's key and important in the early development of blood cells, and hence we have blood vessels coming into it. Eventually it gets absorbed. It's not as important later on in development. But notice that you've got the building up of this heart. Okay, so you're going to have an anterior cardiac vein, that's from the cephalic region. You're going to have a posterior cardinal vein, that's from the caudal region. Okay, and you can see the dorsal aorta here and the aortic arches here. That's 4 weeks. Now, pulmonary circuit carries deoxygenated blood from the right ventricle to the lungs and returns oxygenated blood to the left atrium. Remember I said this a while back, don't get confused with the colors, because colors are used for a diagrammatic purpose only. And what you see in the pulmonary circuit is the reverse of what you see in the systemic circuit. The pulmonary circuit, the arteries are going to be deoxygenated, the veins are going to be oxygenated. In the systemic circuit, the arteries are oxygenated, that is oxygenated blood, and the veins are deoxygenated. Now, staying with the pulmonary circuit, large arteries are called trunks. The pulmonary trunk curves over the superior border of the heart and gives rise to the left and right pulmonary arteries. The pulmonary arterioles provide blood to the alveolar capillary network that surrounds alveoli. Alveolar walls are thin enough to permit gas exchange. The capillaries enter venules, and the blood then enters the pulmonary veins. As you can see right through here, and we will go through this, the general patterns of blood vessel organization. We've mentioned how these are. I encourage you to go over this. Here you see this a little bit more sophisticated. Okay, blood comes back in, gets pumped, and goes out. And there is this trunk here, which goes into the left and to the right pulmonary arteries. And then you have, of course, they eventually go to arterioles and eventually into capillaries. And here in this alveolus, you have gas exchange, oxygen going to the blood, carbon dioxide leaving the blood. That sounds wonderful. Then you have the blood coming back. It is on these veins that are oxygenated, comes into the left atrium, goes through the left ventricle, gets pushed out the aorta. Here's the aortic arch. And here you have the aortic branches there. Okay. Now, the arteries and veins of the systemic circuit operate in parallel and have major vessels often, and the major vessels also often have similar names. I'm going to have you start reviewing the function and location of the systemic artery, starting at the aorta. You need to, if you follow backwards, the flow of systemic veins, starting at the superior and inferior vena cava. One of the ways to study this, and I'm just suggesting this as one real serious suggestion if you want, take an imaginary drop of blood, start at the right atrium, go through the heart, go through the lungs, pick an organ in the body and go from the left atrium, left ventricle, out all the way through that blood system till you get to that organ. That's how you're going to learn. Every one of these arteries and veins that I have mentioned in this PowerPoint, you are going to be responsible for on the exams. Now, why are there both superficial and deep veins in the neck and the limbs? The distribution of veins differs in the neck and limbs as compared to the arteries. The arteries are located deep beneath the skin, protected by bones and surrounding soft tissues. The neck and limbs generally have two sets of peripheral veins, one superficial and one deep. The dual venous drainage is vital for controlling body temperature. In hot weather, venous blood flows through superficial veins, where heat loss can take place. In cold weather, blood is routed to the deep veins to minimize heat loss. Now, as you see one of these diagrams, and you'll see more of these, you want to be clear about what are the arterial arteries and what are the veins, and where are they located. Same thing here with the veins, and you would follow them up till eventually you get to the heart. So the branches of the aortic arch supply structures that are drained by the superior vena cava. So the arteries include brachiocephalic trunk, internal thoracic artery, left common carotid artery, left subclavian artery, vertebral artery, axillary, deep brachial, ulnar collaterals, radial, ulna, deep and superficial arch, and digital arteries. As you look here, you start off with the aortic arch, and here you're going to have the left subclavian, the left carotid, you will have the brachiocephalic trunk, then you will have this go the right subclavian artery, and then you'll follow through with the axillary, the deep brachial, the brachial, the ulnar collateral arteries, the radial and ulnar, and eventually you have the deep palmar arch and the superficial palmar arch, and off of them branches into the digital arteries for each one of the respective fingers. Now with the veins, in the same situation, you want to be mindful of the brachiocephalic vein, the superior vena cava, internal and external thoracic vein, jugular vein, the internal and external jugular vein, the vertebral vein, the right subclavian vein, the brachial, the basilic, the axillary vein, the cephalic vein, the median cubital vein, the ulnar and radial vein, and the deep and superficial palmar veins, the median antebrachial vein, and the digital veins. What are we talking about here? Start from the digital veins. Now, by the way, if you notice, there's a color coding right here. Superficial vein is going to be sort of a light blue, and this is going to be a much more deeper blue for the deep veins. So you can see that there is this network of superficial palmar arch, and off of that you are going to have digital veins. The blood is going to flow back. You're going to eventually also have to deal with your deep palmar arch, ulnar, basilic, radial, okay, cephalic, and then you have these coming together. There's going to be a superficial vein and a deep vein. The brachial is the deep, the basilic is the superficial. All the blood is heading back toward the heart, so keep that in mind. This is venous flow. You will have to also keep in mind the axillary and the junction of the axillary and cephalic, which comes up to be the right subclavian vein here. Note that you'll still have the external jugular, the vertebral, the internal jugular. We'll get into those of the neck leading up into the head in a minute, as well as the brachiocephalic, in which leads into and empties into the superior vena cava. Notice that you also have an internal thoracic here, which also empties into the superior vena cava. Now, the external carotid arteries supply the neck, lower jaw, and face, and the internal carotid and vertebral arteries supply the brain. So you need to be aware of these: the internal carotid artery, vertebral artery, common carotid artery, the carotid sinus, the external carotid artery, and the branches, including the superficial temporal, maxillary, occipital, facial, lingual, and basilar. Here you see this. The common carotid then branches off. You have also the carotid sinus, which is part of the internal carotid. You have the external carotid here, which will branch into lingual, facial, occipital, maxillary, and the superficial temporal. Okay? Notice that you have a branch off of here, the vertebral, following up along the vertebrae. Okay? And you have, of course, the axillary, the subclavian, and the brachiocephalic here. Notice the location that gives you very nicely where the clavicle plays a key role right here. You get everybody that sometimes, if you remember in your bones, the clavicle is one of the most common bones that are broken. Sometimes people ignore that. Oh no, I'll be fine, even though it hurts like all heck. You don't want something that's got a clear break because look how close it is to a major artery. Okay? You don't want something hanging around like that. Now, if you move to the external jugular veins, they drain the region supplied by the external carotid arteries, and the internal jugular veins drain the brain. So the veins you need to be aware of are the external jugular vein, formed the maxillary and temporal veins, as well as the internal jugular vein, which drains blood from the cranial sinuses. You have the vertebral vein, the external jugular branches, the temporal, maxillary, facial, and occipital. As you can see right here, notice again the same situation with the veins, superficial and deep. Dark blue is for the deep, powder blue, the light blue is for the superficial. You have the dural sinuses draining up in here with the confluence of the sinuses right here. Then you follow the dural sinuses until they eventually come out for the internal jugular. Also, which drains from, or I should say, into the internal jugular include the facial, the maxillary, and you can see this also. Now, the temporal, it's a toss-up because it can also drain into here, but the temporal drains into the external jugular, all right? Notice that all of this is passing through the jugular foramen. Here you have also another emptying here of the vertebral, and they all join together at the right brachiocephalic right here. Okay. And of course, you've got your axillary, right subclavian, and superior vena cava. The internal carotid arteries and the vertebral arteries supply the brain. It is absolutely critical. Now, the circle of Willis, I expect you to know. Cold, hard, fast. You have in these arteries the internal carotid artery branches of the ophthalmic artery, anterior cerebral artery, middle cerebral artery, and the cerebral arterial circle, the circle of Willis, as well as the anterior communicating, the anterior cerebral, the posterior communicating, and the posterior cerebral, as well as the basilar, vertebral, and cerebellar, which you can see right here. You have the internal carotid going up. You have the ophthalmic, the anterior cerebral, the middle cerebral. Of course, you've also got from the vertebral coming up, which eventually joins up with the basilar, and eventually comes into the posterior cerebral. But if you look underneath, this is where it gets really describing, as you can see this circle of Willis here, consisting of anterior communicating, anterior cerebral, posterior communicating right here, the posterior cerebral. You have the basilar delivering here from, and it comes up from the vertebral. Notice that you've got the cerebellar right here, okay? And you have, of course, the posterior cerebellar. Notice where the pituitary gland is sitting, right around here. So it's a navigation guide here. The middle cerebral. Now what they did was they did a cut here of the internal carotid as it comes through. You have the ophthalmic, and you also have the anterior cerebellar right here. Okay? Now when you talk about venous supply, you want to keep in mind that this is all drained by the dural sinuses and the internal jugular veins. You have unpaired cerebral sinuses, which are the superior sagittal sinus, the inferior sagittal sinus, and the straight sinus. You have a cavernosus sinus, which drains into the petrosal sinus. You have paired lateral sinuses, the right transverse sinus and the right sigmoid sinus. The petrosal sinus drains into the transverse sinus, and then you have the vertebral vein, and the confluence of sinuses empty into the internal jugular veins. So here we have the superior sagittal sinus here. This is taking in blood. You have the superior sagittal, inferior sagittal right here. So think of superior, it's above here. This one's inside. straight sinus right here, cavernosus sinus right here. This is the confluence of sinuses, meaning 1, 2, 3, they're all coming together. Then you have the right transverse sinus here coming across right, the right sigmoid sinus here, and the petrosal sinuses, which are here and here, emptying into the internal jugular, and this one, the occipital sinus, empties into the vertebral vein. Okay. When you take a look at this from the underside, you see the superior sagittal sinus is cut. You have the cerebral veins here, here, here. The petrosal sinuses here and here. The sigmoid sinus here. This is a transverse sinus here. The confluence of them all is sort of in the back, okay? But it is below the cerebellum. You have the cerebellar veins here and here. And you have the internal jugular here. And you have one here, okay? And this is the cavernosus sinus right here. Now, regions supplied by the descending aorta are drained by the superior and inferior venal cava. What we're saying is that you deliver the blood from the aorta, and eventually it will go and be recollected by the different aspects of the vena cava. But when we talk about the arteries, we're going to talk about the intercostal arteries, superior phrenic arteries. Phrenic should give you a tip off that that's going to be involving the diaphragm. The bronchial arteries, esophageal arteries, mediastinal arteries, and the pericardial arteries. You want to note that there are paired branches and unpaired branches of the abdominal aorta. So moving from the aortic arch here, we start going down. You will have the visceral branches of the thoracic aorta, which include the bronchial, esophageal, mediastinal, and the pericardial. This is the dividing line, folks, to keep in mind. This is your diaphragm, okay? As a result, you have the intercostal arteries here. You have the superior phrenic, which kind of sits on top of the diaphragm here. Then you're going to start dealing with the celiac trunk, and you will have branches off of it, the left gastric, the splenic, the common hepatic, as well as a major branch, the superior mesenteric, and then a lower one. Now the superior mesenteric is right here. The inferior mesenteric is right here. The abdominal aorta is right here. And what defines it is really the diaphragm. Once you pass from here to here, okay, you've gone from thoracic aorta now to abdominal aorta, okay? Then you will have this branching off at the common iliacs, okay? You will notice also that because you have a pairing of organs, such as kidney to kidney, adrenal to adrenal, etc., there will be a pairing off of the various branches of some of the arteries from the abdominal aorta. These branches will include the adrenal, the renal, the gonadal, okay? You've got some of the branching of the lumbar, and by the way, we will also have, as I've shown you where the diaphragm is, there will be an inferior phrenic, which is on the underside of the muscle, the diaphragm. And so what happens is when you get to the abdominal part, you're going to have major paired branches, the inferior phrenic, adrenal, renal, gonadal, and lumbar. Then you will have major unpaired branches, meaning that there's just one. The celiac trunk, the superior and the inferior mesenteric arteries. Now, the regions supplied by the descending aorta are drained by the superior and inferior vena cava, as we mentioned. These include, for the veins, the azygos, the hemiazygos, and these veins receive blood from intercostal veins, esophageal veins, bronchial veins, mediastinal veins. These are the major branches of the inferior vena cava. Starting right here, you can see where The azygos is, all right, and you have the branches of the mediastinal, bronchial, and esophageal veins. You also have the intercostal veins. And coming up here, you have the hemiazygos, which delivers into the brachiocephalic, as well as the zygos, which will eventually empty into the superior vena cava. You have the internal thoracic here, and again, you're still at the inferior vena cava. Once you go below the diaphragm, you're in a completely different area now. This is all the abdominal, so you're going to have the phrenic, you're going to have the pairing off of the adrenal, the renal, the gonadal, the lumbar, and of course then you're going to have the common iliac. Okay, so you see this here listed. Now the visceral viscera are supplied by the ciliac trunk and the mesenteric arteries and are drained by the branches of the hepatic portal vein. Now, 3 unpaired branches, arteries, celiac, common hepatic artery, left gastric artery. We've got the splenic artery. Now, those are all branches off of celiac trunk. The superior mesenteric artery, you have the inferior mesenteric artery, and then we have veins. The hepatic portal vein fusion is from the superior mesenteric vein, the splenic vein, the inferior mesenteric vein. What you absolutely have to know, dead flat on top of it, is the hepatic portal vein. This is unique. It directs blood with absorbed nutrients from the digestive system to the liver for processing. And what you see here, let's start off with this. This is the celiac trunk, which will have the common hepatic artery to the liver. It will have a left gastric artery, which is to the stomach. It will have a splenic artery, which eventually delivers blood to the spleen. Branches of it will also include one side of the stomach as well as the pancreas here. Then we have the superior mesenteric artery. Now, you don't have to know every one of these little guys here, but you do have to know how the superior mesenteric artery is an unpaired artery that takes care of a certain proportion of small intestine, large intestine, okay, and to a point, the pancreas and the duodenum up in here, okay? Now, the other major unpaired artery is the inferior mesenteric artery, which goes into and feeds to the colon and to the rectum, okay? Now, let's talk venous, the venous supply. Here's where you've got to keep in mind. Remember, the venous is bringing blood back through. Now, here's the thing. We've got the inferior vena cava here, okay? We have absorbed a lot of nutrients. You have the left and the right gastric. They're both going to empty into this hepatic portal. You have the pancreatic, and you've got the inferior mesenteric vein, which is taking nutrients, etc. It's also going to be taking it from the colon and the rectal. The thing to think about is this. What's the difference between superior mesenteric and inferior mesenteric, whether we're talking artery or vein? Superior is above. It is going to handle a lot more of the small intestine, some of the large intestine, but also the pancreas, duodenum. Okay? Inferior is a stretch. It handles more or less the lower part of the colon, and that will include also the rectum. All right? But what is all this stuff doing? It's going over to the hepatic portal, which will then distribute all of the nutrients, and it will take away certain materials. But guess what? All of this is going to go over here, and then it's going to be picked up by the hepatics and deliver finally into the inferior vena cava. Okay, so anything that leaves here has to still go through the liver, and then it will get into the inferior vena cava. All right, So I've mentioned these: inferior mesenteric, splentic vein, okay, and superior mesenteric vein. The pelvis and lower limbs are supplied by the branches of the common iliac arteries and drained by branches of the common iliac veins. The artery. Right and left common iliac arteries. They're elastic. They have to be because they take a lot of variations in pressure. But these are divided into internal iliac, femoral deep femoral, femoral circumflex, popliteal, posterior and anterior tibial arteries, and then the anastomosis is around the dorsalis pedis and the dorsal arch and the plantar arch, as you see right here. Handling all the pressure and all the blood supply now going through the pelvic region, internal iliac, starting with the common iliac, the external iliac, the internal iliac, down the femoral, deep femoral is a branch, femoral circumflex, okay? Descending, we're going to the popliteal, anterior posterior tibial, and then the fibula, and then we have the arteries of the foot, okay? You can see the back end of this where the fibular comes down, and they basically have the arches here. The plantar arches, medial and lateral, and the dorsal arch, and the plain old plantar arch, okay? Now, what about the veins? The pelvis and lower limbs are supplied by branches of the common iliac arteries and drained by branches of the common iliac veins. Yes. So, in the veins, we're going to be talking about the common iliac vein, external and internal iliac, the femoral, the great saphenous, the femoral circumflex, the popliteal, small saphenous, anterior and posterior tibial, and in the fibular, as well as in the foot, you will have the plantar venous arch and the dorsal venous arch. Okay, here's the thing. The plantar venous arch is here. The dorsal venous arch is here. And of course, you've got the digital veins right there. You're going to come up with the fibular. You have the anterior and posterior tibial. See where they're located. The small saphenous, and then you have this rise up until the popliteal. Now, if you notice this big, large vein here, the greater saphenous, I may have mentioned it in the lab, but I'm going to mention it again, just to remind you. That vein is one that there's a section taken out if they have to do a cardiac arterial bypass, or arterial graft, as they would call it. They take out that piece of that vein, and they basically do the bypass around where the arterial, the cardiac arterial vessels are blocked by clots. And seeing that I had this with my mother-in-law, that was one of the coolest things, because she had a triple, almost quadruple bypass. And basically 4 clots, or almost 4 clots, and they basically bypassed the blood flow. And she was up and around in so many days and giving me grief like good mother-in-laws always do. Short time after that, but no, God bless her. It was great. But it's the thing that you have to understand. You just take tissue from your own body, take this vein, a piece of it, and graft it onto the arteries to bypass the blockage, and the heart continues to function very nicely as a result. Note that you have the deep femoral, the circumflex, you have the femoral, and then you have this larger structure, the external and internal iliac, as well as the common iliac. Now, the arteries of the systemic circuit deliver oxygenated blood through the body. I want you to review that. Note, bronchial arteries supply the tissue to the tissue of the lungs. They are not involved in gas exchange. Okay, so bronchial arteries aren't involved in this pipeline of taking deoxygenated blood bringing it up to the alveoli, taking it away to be delivered back to the heart. They're separate. This flow chart will give you another approach if you have some difficulty. Same here and here. Okay? The veins of the systemic circulation return deoxygenated blood back to the heart. And that's what I want you to review here in this flow chart, and here, and here. The pattern of blood flow through the fetal heart and the systemic circulation must change at birth. Keep in mind something. Fetal blood flow is occurring to the placenta through a pair of umbilical arteries and a single umbilical vein. Now, when they have the delivery of the baby, they always will check the umbilical arteries and the umbilical vein. It's always 2 arteries, 1 vein. If there's any difference in that number, then that tells the obstetrician and the doctors immediately there may be a problem. Okay? Why is this so important? Because in part, we're having a bypass in the heart. See, if you're inside of mom's womb, you're not going to be able to breathe on your own. So therefore, you don't have as much of the necessary blood flow to the lungs. The blood flow to the lungs is set primarily to allow for pulmonary structural development only. No gas exchange. Gas exchange occurs at the placenta. Okay? But you have several bypass structures that occur in the heart to move blood through the heart, then back to the systemic circulation, and eventually it will get sent out on the umbilical arteries for gas exchange again. So you have to see the structures of the foramen ovule, which eventually develop into the fossa ovalis after birth. This is the little hole that shuts close and eventually fibroses over. If it doesn't, then surgeons have to go in and close it up for the infant. The same thing with the ductus arteriosus. Now, the ductus arteriosus will close due to pressure changes, and eventually it fibroses up, and that's the ligament arteriosum that we see. So here you have the blood flow going through that point of the foramen ovule that will close. You have this bypass that occurs of the blood, and that is the ductus arteriosus. Okay, this is the pulmonary trunk, and it's a bypass that eventually gets blood flowing. Notice that where the blood will come up to the umbilical arteries, they will join together and be wrapped around the umbilical vein. At the umbilical cord, it's the connection with the placenta, where the the fetus is getting gas and nutrient exchange with mom's circulation. When birth occurs, of course, we cut off the umbilical cord. There is also going to be that first breath. When the baby takes that breath in, there's a pressure change in this thoracic cavity that causes these structures to pop close. Also, we have the ductus venosus, which itself will also eventually atrophy and close up. Now, we've been talking about some of this, and you're seeing up close here. Here is the ductus arteriosus, it's closed. Here's the foramen ovule, it's now been closed. And the blood flow comes in through the superior and inferior vena cava from the right atrium into the right ventricle, out to the pulmonary arteries for gas exchange, back to the heart, to the left atrium, then out, okay? This is after delivery. Now, there are several defects that I want you to be aware of. You need to know all of them. First one is a septal defect. In this ventricular septal defect, this is where this ventricular septum, this interventricular septum did not completely form. Hence, what do you have? You have a mixing of oxygenated and deoxygenated blood. This is going to lead to problems of proper oxygenation for the infant. You have also what is called a patent foramen ovule or patent ductus arteriosus. In other words, they're patent, they're kept open. These things didn't close. These can be surgically set, doesn't take long. It is considered open heart surgery, but as you notice here, you have oxygenated blood mixing with deoxygenated blood, and so there's an inefficiency in the blood flow and the oxygenation to the tissues. Now, this is called tetralogy of Fallot. Now, tetralogy, tet means 4. So in this case, you have 4 developmental defects. The patent ductus arteriosus. You have a pulmonary stenosis. Now, the stenosis is a narrowing and hardening. So this valve is not opening as fully and completely as it should. You also will have a ventricular septal defect here, again, mixing of oxygen, deoxygenated blood, but also you'll notice that the thickness of this heart wall has been enlarged. Why? Because it has to get much more muscle to force blood through this stenosis, this narrowing of the pulmonary trunk. So what will happen here is they will have to close off the wall here, close off the bypass here, open or widen this somewhat, this pulmonary stenosis here. In doing so, that will probably reduce all of the defects and will allow the heart, the right heart ventricle to eventually atrophy a little bit, but then get into the proper proportion. Okay, remember, the bigger, thicker muscle wall will be on the left ventricle normally. Okay, you have an atrioventricular septal defect. Now, in this case, here's the atrial defect, which is going and mixing oxygenated blood with deoxygenated blood, atria to atria. And so that septum has been messed up. You have now a defect there that has to be closed up, as well as the ventricle. So you now have, even when the ventricle contracts, it's mixing the blood that is oxygenated with the deoxygenated, so it becomes very inefficient. Okay. And finally, the transposition of the great vessels. This is sad. It's a little bit more of a challenge, but it can be dealt with. One, you still have a patent ductus arteriosus. That has to be closed. Here is your aorta. Here is your pulmonary trunk. Translated, right side is delivering blood to the left function, which would be systemic circulation. So you get basically deoxygenation. Left side is delivering blood. Some of it leaks back to keep the infant having some tissues oxygenated, but it's completely a mess because it's supposed to be this side delivering blood to the pulmonary arteries and this side delivering to the aortic arch and to the rest of the body. So it's a greater reconstruction of the heart that occurs. Okay, we've pretty much dealt with everything, and I think that's wonderful. I want you to encourage you to peruse chapter review outline, which is pages 743 to 744, 743 to 747. Complete the review questions on 748 and 749. Complete the review sheets on pages 704, 720, and 742. In each case, remember, these are not assignments that I'm going to have you deliver back to me. Don't do that. This is to give you further review tools to help you to prepare, to give you also assurances. Do you know what you're supposed to know? And if you come up and say, gee, I'm stumped, then that means at a particular section, you've got to review that material. Also, the next lecture will be on Chapter 21, the respiratory system. And I hope you all have a nice day.